So good afternoon. Is it audible? Okay. Fish pollen. Hmm. Okay. Very good afternoon, everybody. Now we have discussed the anther and pollen culture. Anther and pollen culture. In adjacent, the haploid and gametophytic phase is extremely brief and represented by the pollen grains in anther, and one or more cells in embryo sac of an ovule. So, typically, anther will take the cross section here, the two anther lobe, and each lobe possesses two pollen sacs. So, when the microsporogenesis, pollen mother cells inside the pollen sac from pollen tetrad uh, by the occurring the meiosis division. In each pollen tetrad, four pollen grains are there, which is together temporarily by their callus wall. The pollens are separate as a discrete unit by the dissolution of callus wall. Each pollen possesses an unique genome where every gene is present as a single copy. We know that one. So exploitation of this unique genetic unit and the totipotency of plant cells is the basis of anther or pollen culture for the production of haploid plants. So mainly the pollen grains to produce would the haploid plants only. Okay. On the other hand, egg cells produced within the ovule is very difficult to separate from complex tissue integrated. In the culture of these anther cells and dehiscence along with its upper margin lengthwise, we are take that one. This phenomenon helps to expose the pollen grains alternately. High amount of the pollen grains can be isolated manually and the culture receptacle very easily. The pollen grain is more suitable material than egg cells for the production of haploid cells, means haploid plants. The development and production of haploid plants in vitro is very important for the study of fundamental and applied aspects of generic in higher plants. The production of homozygous diploid by doubling chromosome number of applied in vitro marks to a pure line in the single step, and such homozygous pure lines is a great importance in the plant breeding. So, from the practical as well as fundamental point of view, the anther and polyculture is upon a new avenue in the field of plant breeding or plant sciences. So, what about the brief history of these anther and pollen culture? Quickly, so Tulichi is the first observed that mature pollen grains of ginkgo biloba. It can be introduced to proliferate culture to form the haploid callus. After that, Guha and Maheshwari in 1964 first is reported the direct development of embryos from microspores of the Datura plant. So after that, Nietzsche and that is Borgin, the obtained the complete haploid plant from the anther culture of the Nicotina tabacum. Okay. What is anther culture? The anther culture is the technique by which is the developing anther at the precise and critical stage are excised aseptically from unopened flower buds and are cultured on the nutrient media where the microspores within the culture anther develop into callus tissues or embryoids that give rise to haploid plantlet either through organogenesis or either embryogenesis. What is the pollen culture? The pollen, either microspores, the culture in vitro technique by which the pollen grains, preferably at the uninucleus stage, are squeezed out aseptical from the intact anther, then the culture on nutrient medium, where the microspores without the producing male gametes developing into haploid embryoids are callus tissues that gives rise to the haploid plantlets by embryogenesis or either organogenesis. What is androgenesis? Next, here one by one we have to discuss that one: anther culture, pollen culture, and androgenesis. The androgenesis is the in vitro development of haploid plants originating from the totipotent pollen grains through a series of subdivision and different There are two modes are there to the androgenesis, that is the direct androgenesis or indirect androgenesis. Direct androgenesis, either another one is the indirect androgenesis. Direct androgenesis, the microspores behave like a zygote and undergoes changes to form the embryoid, which is ultimately gives rise to plantlet. Here there is no formation of callus. So directly the zygote to develop as a plantlets, that is the direct androgenesis. In indirect androgenesis, what happened here? In the contrast to the direct androgenesis, the microspore instead of undergoing to embryogenesis divide repeatedly to form the callus, which is differentiated into applied plantlets. This is the indirect androgens. This is the anther culture. See that shown the diagram here. The principle of anther and pollen culture. Here, what are the principles are there? We have to discuss here. The basic principle of anther and pollen culture is production of applied plants, exploiting the totipotency of microspore and the occurrence of single test chromosome. So in this process, the normal development and function of the pollen cells to be the male gamete is stopped and mm, diverted forcefully to a new metabolic pathway for the vegetative cell division. For this objective, the microspores either within the intact anther or in the isolated stage are the grown aseptical on the nutrient media where the developing the cell grains means the pollen grains will form the callus tissues or embryoids that ultimately gives rise to applied plantlets. In fact, so the anther culture is sense to pollen culture. Yeah. In culture condition, the diploid tissues of anther will remain living without proliferation at the selective medium. At the same time, it will encourage the development of pollen by nourishing and providing the nutrient media. The haploid embryos or the callus tissues can be seen as antheridaceans in the culture, but there is always the possible that the diploid somatic cells of the anther will also respond to culture condition and produce unwanted diploid cells, or that one we call as the plantlets. In attempts to avoid these problems, free pollen isolated from the anther are grown in nutrient medium. The knowledge again so far from anther and pollen culture has established that pollens at the uninucleate stage just before the first mitosis, during the mitosis, the most suitable for the induction of haploids. Introduction of hoplites can be enhanced by the keeping the anther flower buds at the low temperature. The low temperature has been ascribed to a number of factors such as dissolution of microtubes, alternation in the first mitosis, maintenance of higher ratio of viable pollen capable of embryogenesis. Could the treatment may also act to the help the embryogenesis by representing the gametophytic differentiation or by the lower or abscisic acid content at the anther, which is considered to be inhibitory for the production of applied plants. The important aspect of anther and pollen culture is the nutrient media. Mainly, the nutrient media they have the growth hormones and uh, micronutrients and agar-agar, all the minerals which are suitable for the growth of plants. Then that is the nutritional requirement is exist anther and much similar than those isolated microspores. In the isolated microspore, it is obvious that certain factors is responsible for the induction of haploids, which might have been provided by the anther are missing, and these have to be provided through the medium. The rich medium may encourage the proliferation of the diploid tissue of anther, that anther wall, and should be avoided. The incorporation of activated charcoal into the medium have the stimulated induction of androgenesis. The iron in the medium also plays the important role in the induction of haploids. Potato extract, coconut milk, and growth regulators like auxins and cytokinins also used to the anther and culture, pollen culture due to their stimulate the effect on androgenesis. So that is the coconut milk and all are the is very essential for the growth of the anther culture or in the flora. So these are the very essential. In culture, pollen may be divided mitotically, can follow the normal pathway for forming the vegetative or generative cell. The generative cell, that is generative nucleus, which is quiescent and about the vegetative nucleus divide repeatedly forming the multinucleate in the pollen grain. The multinucleate pollen is undergoes segmentation, which may be used to form the either organized embryonic structure and callus tissues. The haploid plants are self-sterile due to the presence of a single set of chromosomes which are not able to participate in the meiotic segregation. By Colchin treatment, the haploids are made homozygous diploid or isogenic diploid, which are fertile condition is present. The haploids are homozygous diploid grown in vitro are transferred to pot and grown to maturity in the glass house. So protocol for anther culture. The tobacco is the ideal material for anther culture. So the basic protocol described below should be applicable to anther culture in general with the modification. The immature anther containing the uninucleate. अरे इटा घुसोर so the basic protocol is described below should be applicable to anther culture is general with the modification. The immature anther is containing the uninucleate pollen at the time of first meiosis, means mitosis, are the most suitable material that is the in the production of haploid. So some steps are involved when they are to undergo to the that is pollen means anther and pollen culture. The collect flower buds of Nicotina tabacum at the one set of flowering means normally in 3 to 4 months old plant means the maturity but not the essence flowers. Measuring the length of each individual with the in centimeter, the selection of flower buds around 17 to 22 centimeters in the length and sepal equal at the petals. So that reject the at the all the flower buds which are beginning to open. So transfer of selected flower buds to the laminar airflow. Each flower buds containing the 5 anther and these are normally surface sterile in the closed buds. The flower buds are surface sterilized by immersion in 70% ethanol for the 10 seconds are followed immediately by the 10 minutes in 2% of sodium hypochlorite. There are washed these three times with the sterile distilled water. Finally, transfer to the birds a sterile petri dish. See here, this diagram is already there. So here, see that protocol I shown to that figure. Hmm. To remove anther, slit the side of birds with a sharp scalpel and remove them with a pair of forceps. Place the five anther with the filament to anther petri dish. The filaments are cut gently during the extraction of anthers. A special care should be taken to ensure that they are not injured in any way. Mm, the damaged anther should not be discarded as they often from the callus tissue from the damaged part other than the pollen. The anthers are placed on the agar sulfide basal MS or white and or niche medium. The culture is kept into initially in the dark after 3 or 4 weeks. The anthers normally undergo pollen embryogenesis and haploid plantlets appear from the culture anther. In some times, the anther may undergo proliferation due to the form of callus, which can introduce the differentiate into haploid plants. At this stage, the culture is incubated at room temperature, means 24 to 28 degrees in 14 hours. Daylight region. Um, the approximately 500 the tall plants are freed from agar by gently washing with the running tap water and transferred to small pot containing the autoclaved putting compost. Cover each plantlet with a glass beaker to prevent the desiccation and maintenance in the well humidity and greenhouse. Group and after 1 week, remove the glass beakers and transfer to the large pots when the plants will be mature and finally flower. This is the see here. This one is the diagram is shown the anther and isolated microscope, the developing applied plant directly by the embryo formation to the applied culture. Coming to the protoplast culture, so protocol for the pollen culture. So isolated pollen can be cultured by two methods are there. This method is described here for the culture of isolated pollen of tobacco. This technique is considered as the basic protocol for the pollen culture and involve the following some steps are there. What is the steps here? Hmm. The selection of suitable unopened flower buds, sterilization, excision of anther without filament are the same as described previously in the anther culture. About 50 anthers are placed in the small sterile beaker containing 200 ml of liquid medium, that is the nutrient medium. Anthers are then placed against the side of the beaker with the sterile glass of the syringing to squeeze at the pollen. The homogenized anthers are filtered thoroughly in the nylon sheaths to remove the anther tissues, that is the anther debris and tissues are there. The filtrate of or either pollen suspension is then centrifuged at the low speed for 5 minutes. The suspendent supernet content fine debris is discarded, and the pellet of pollen is suspended at the fresh liquid medium and washed twice by repeating centrifugation and precipitation in the fresh liquid medium. The pollens are mixed finally with the measured volume of liquid basal medium, so that makes then here around 10 to the power of 3 or 10 to the power of 4 pollen grains in that medium. The petri dish, which are incubated at 27 to 30 degrees under the low intensity of light, cool, white cool light. The young embryos, that is embryoids, can be observed after 30 days. The embryoids ultimately gives rise to the applied plantlets. The haploid plantlets are then incubated at 27 to 50 degrees daylight regime at about the 200 logs. So the transfer to the soil, as I described already, we are in the anther culture. So next method two, this method is known as nurse culture technique, which is developed by the Sharp et al. In 1972. So here the selection of here steps are there in these nurse method. The selection of flower buds, sterilization, excision of anther, isolation of suitable pollen are same as described in the already in the method one. In this method, the intact anther are placed horizontally on the top of solid or semi-basal, semi-solid basal medium means semi-solid nutrient medium in the conical flask. A small filter paper disc is placed on the intent anther and that about 10 pollen grains in the suspension and then placed on the filter paper disc. Hence, these intact anther are considered as nourished tissues. A control set is also prepared is exactly the same way, except that the pollen grains and the filter paper is directly kept under the solid medium. Sometimes the callus is derived from any part of the plant. It is used as a nourished uh, tissue. With this method, the pollen grains is controlled so it did not grow at the all. The pollen grains kept on nourished tissues grow and form a culture of green parenchyma tissue is after 2 weeks. This is the some of the uh, pollen culture. Okay, so pollen culture and some protocol of the pollen culture. Here, two methods are there. One is the direct method, another one is the nurse method is already discussed. So next one is the advantages of pollen culture over the anther culture. What are the advantages? The pollen culture is five. It has been already proven that anther culture is the efficient way for the production of applied plant from the microspore present with the intact anther. In this process, there is always the possibility that somatic cells of the anther that are applied means that are deployed, which will be represent the respond to the culture condition and produce unwanted deployed callus or plantlets. Sometimes development of microspore inside the anther may be interrupted due to the growth inhibiting the substance leak out the anther wall in contact with the nutrient medium. In the attempts to avoid these problems, the culture of free pollen has been investigated. The culture of of the pollen offer following the additional advantages are there. The pollen culture is also in the advanced condition, that is the overcrowding of pollen grain in anther is eliminated and isolated pollen grains are equal exposed to nutrient medium. Unwanted growth of anther wall and other associated tissues are eliminated. The steps of androgenesis can be observed starting from the single cell. The various factors governing androgenesis can be better regulated. Pollen is ideal for uptake and transmission and mutagenic studies, as pollen can be uniformly exposed to chemical and physical mutagens. The higher the various factors growing the androgenesis can be better regulated. Pollen is ideal. The pollen may be directly transferred to the embryoids, so it is very suitable for understanding biochemistry and physiology of androgenesis. Higher yields of haploid plants per anther could be uh, expected in the pollen culture than the anther culture. So this is a very fun. So another diagram is also there that means the picture for the growing the here. Hmm. Homozygous diploid plants, they can put it homozygous plants. The haploid plants is derived from either anther culture or pollen culture. These plants contain only one set of chromosomes. By doubling their chromosome number, the plants can be made fertile and resultant plants will be homozygous diploid or isogenic diploid. These homozygous diploid plants show the normal meiotic segregation. The fertile homozygous diploid plants are more important than the sterile haploid plants. Homo- Homozygous diploid plants can be used as pure lines in the plant breeding technique. So the haploid can be diploidized by the different methods are there. So here colchicine treatment also one of the method to haploid plants to develop as a diploid. The colchicine has been utilized widely as a spindle inhibitor. Spindle fiber inhibits, so the induced the chromosome number will be diploid. Duplication forms are occur. So here This method has been employed for the obtaining the homozygous diploid the plants from haploid culture. The young planted while still enclosed with the anther are treated with the 0.5% of oxygen isolation for the 24 hours. The treated plantets are replanted in the medium for after throughout the washing. In case of mature haploid plantlets, 4% of calcium. So lanolin, the paste may also help for the axil of leaves. Another method is there endomitosis. Endomitosis also. This one is the haploid cells is unstable in the culture and have the tendency undergo to the endomitosis. That means the chromosome duplication without nuclear division. This property can be used for obtaining homozygous diploid plants. In this process, a small explant of stem from the haploid plantlet is cultured and axin cytokinin iodide medium where these segment from the callus tissues. During the callus growth, diploid homozygous cells are produced by endomitosis. Now, large number of isogenic diploid plants can be obtained by organogenesis. So, coming to the another one is the fusion of polynuclei. This is also to produce the that is diploid plants to encourage the diploid plants. Homozygous diploid callus or embryoids formed by the spontaneous fusion of two similar nuclei of the culture pollen after the first division of that is the mitosis. That is also there. So another one is the significance of haploid plants. What are the significance of haploid plants? Here in the diploid cells, the chromosomes exist in homolog- homologous pairs. The genes for specific characters are also formed in the pair which are known as allelic gene pairs. So that's why here this one is a chromosome contain the number of dominant and recessive genes. Those allelic forms are present on the homologous partner of chromosomes at the same gene loci. In heterozygous diploid, only the dominant allele are expressed phenotypically. The homologous chromosomes is separated during the meiosis. The pollen grains is recessive only on the set of homologous chromosomes. As a result, the pollen-derived haploid plants, all the recessive genes along with the dominant genes will be expressed phenotypically as there is no making recessive genes by dominant gene. So as a result, the dominant as well as the recessive genes will be doubled at their representative loci. So therefore, the comparing the heterozygous diploid plants with the homozygous plants, one can easily to identify the recessive characters which are not possible to identify in the heterozygous condition. So when the crossing over is also essential feature in the meiotic cycle by which is a random exchange of genetic material between two homologous chromatids. The exchange of is a great importance because it is the produce a new gene combination. As a result, the four haploid nuclei produced from a single diploid nuclei difference from one another. Therefore, the haploid plants is derived from four haploid pollen of pollen tetrads. These are these are the significant because the plantlets will be differ genotypically. Why the haploid plants are sterile? Here, this one is discussed later. No need of that one. Another, the importance of and implication of anther and pollen culture. In vitro anther and pollen culture for the production of haploid and homozygous diploid have proved an important tool for the fundamental and applied the plant biology. Haploids are very important for the study of fundamental genetics, the higher plants, because the all the recessive genes are remain unconverted and the recessive genes control traits are expressed phenotypically. Hmm. Regenerated plants is also valuable for the mutation mutational study. In addition, the haploid is very useful for the plant breeding program. The importance and implication of anther and pollen culture is given below the details. The utility of anther and pollen culture for the basic research. What are the here some researches we are to involve? The haploid derived from anther and pollen culture are useful in cytogenetic studies, cytological studies also. By comparing the heterozygous diploid with the haploid or homozygous diploid, population recessive phenotype characters can be identified very easily. Critical genetic analysis of haploid population derived from the individual microscope of pollen tetrads is very useful uh, for the study of genetic recombination in higher plants. The series of cell division and mode of differentiation starting from the single cell and ending with the whole organism can be studied under a microscope. Double haploid that are the homozygous and fertile are readily obtained enabling and selection of desired gene combination. The double haploid that is the means culture of isolated pollen provides a novel experimental system for the study of factor controlling the pollen embryogenesis in the higher plants. So another one is the study of meiotic behavior. Sorry. Uh, Another one is there. What is that one here? Sorry, technical issues are there. The genetic analysis could be performed on the haploid population to establish inheritance patterns. Another application involved the use of haploids in the production of monosomic and lalisomic and other aneuploids. This approach has been used in the tobacco for isolation of nalisomes, trisomic and tetrasomic. Um, some uses of haploids in the uses of sorry in cryogenic study. What are the here cryogenic studies also we have to use. So for the cryopreservation of haploid cells, pollen embryoids and applied meristem, the meristem tips at the super low temperature in the liquid nitrogen of hence a novel approach for the long-term preservation of genetic stability and to establish the applied germplasm blank. The pollen embryos and haploid meristems are genetically stable and develop into entire plant easily. Therefore, these material are more suitable for cryogenic So, use of anther and pollen culture for the plant breeding and crop improvements. So, this one also in the anther and pollen culture is also very useful technique when the breeding and crop improvement techniques. What are there here? The main and the foremost advantages of the in vitro production of haploid over the conventional plant breeding method is the saving of time. Is very it's less time it is to produce the number of plants. By anther and pollen culture, homologous diploid or isogenic diploid plants can be produced within the year to compare the long breeding method. It may be takes 4 to 6 years means conventional breeding method. The isogenic lines are also beneficial where the plantlets are self-incompatibility, for example, ray plants. In inbreeding programs, the isogenic line facilitates facilitates the work in the introducing desirable mutations, the transformation of biochemical mutations. The success of any crop improvement depends on the extent of genetic variability in the base of population. In this regard, the Kailas culture is a rich source of genetic variability by Antar culture, not only applied, but plants have various ploidy level, the mutation will be is regenerates. Uh, and in China, the culture is new varieties of rice, soya, and different varieties are produced. So, for example, Hevia braziliensis, that is the rubber yielding plant, you know that one. It is the perennial crop pollinating tree, and in the available varieties are highly heterozygous in nature. So here 5 to 7 years are required for showing to blooming and seed set ratio of breeding in usually only few ten thousands. So it is unreal to obtain pure line by the means of successful inbreeding, maybe regeneration. The pollen plants have been produced by anthroculture through the embryogenesis. Therefore, the production of homologous diploid pollen plants in a new way to obtain pure lines in the different genotype, very short period and can be utilized in the breeding programs. And the applied means technique to serial breeding is a kick route for the heterozygous to homozygous in serial production. The anther is derived applied have been extensively used in the protoplast culture and somatic hybridization. So application of applied culture for the horticultural plant breeding technique. In some plants of horticultural importance, applied culture is highly significant. For example, Tracia. Tracia is the horticultural important plant, propagates the vegetatively by means of underground ponds. The normally it takes around 8 to 10 years to produce the clone which is longer enough for the commercial purpose. But in these cases, the anther culture can be successfully used to obtain clone very quickly, very short period. We have to produce the clone through the anther or pollen culture. Another application is also anther culture and alkaloid content. Amongst other uses, that is the homozygous plants obtained through the anther culture have also been employed for the selection of breeding lines of Nicotina tobacco with higher alkaloid content. Homozygous recombinants of having the higher alkaloid content, which is obtained by the anther culture only. So another haploid culture in the molecular biology. In genetic engineering, the haploids can be successfully used to gene transfer. The applied tissues of andrographis, Arabinob, Arabidopsis, Arabidopsis, and Lycoperdicus have been used for the transfer of and expression of the gene from the Escherichia coli. So, so this is the pollen and anther culture. So what are the technique here and some applications of these pollen culture and anther culture. Okay. So see here. So another picture I show to. This one is also is there for the pollen and anther culture. Okay. So thank you everybody. Thank you so much for attending the class. Next class we are discussed with the another topic. This one is anthar and pollen culture is very important in the tissue culture topics. Okay. Thank you very much. Thank you everybody.